Can you see? So let's go to the flagella and pili. Okay, flagella, they are made up of three parts. That is basal body, then hook and filament. Basal body. So this is our basal body. This part. Okay, basal body. Then this is hook. Then this is filament. This basal body, right? It is in the cell wall and cell membrane. You can see here, right? This is our cell wall. Suppose this is the cell, right? This is the cell. Two layers will be there, right, for the cells. Okay, here in the middle, right, what will be present? Flagella, right? Here we saw there is flagella present, and that was our magnified image. That picture was of magnified image. Okay, so flagella, where it is in between the cell membrane, right, and also the cell wall, there is flagella. Got it? So let's see here. How many parts? Basal body, then hook, then filament. Got it? So basal body, here it is made up of two pairs of ring. One is outer pair, the other one is inner. So this ring here is outer and this one is inner. Outer pair of ring, what kind of ring they have? L and P. Inner ring, what kind of ring they do have? S ring. S and M. In the gram positive bacteria, right, there will be only one pair of ring, inner ring. Only this ring will be present. This will be absent for gram positive bacteria. We will learn what is gram positive bacteria. Okay. Next, you can see hook. After the basal body, hook will be there. Hook will be present. It is the middle part. Next, we have filament, the outer part. Okay. This filament, right, it is made up of which protein? Flagellin. Okay, it is contractile protein. That means it contracts and relaxes. Because of that, only the bacteria is moving. Got it? Next, we have the pili. We saw, right, this. So this pili, there is two types. One is longer, the other one is shorter. The longer one is called F pili or sex pili. Okay, this sex pili, right? What happens actually is, right, this is the bacteria, and here you see this is the genetic material, okay, of this bacteria. And if it has six pili, what will happen if the other bacteria, if it is not having the six pili, what will happen when they meet? This we already saw, genetic material, and here was the six pili. Okay, when these both meet, because this is not having any genetic material, what will happen? There will be reproduction. And how this type of this reproduction, what will happen? This will form a link. This pili will form a link. I can do this in pink color. What will happen because of that? Both will come closer, and this thing, right, it will replicate here. Here also the genetic material will be formed. And this type of reproduction is called conjugation. This happens in bacteria. There is no meiosis, only mitosis, but one of the ways, right? You have seen there are many different types of bacteria, right? Bacteria become resistant to some antibiotic. So there is some changes in the genetic material. How that changes happen, right? So there should be some transformation of genetic material, right, from one bacteria to the other. So how this is possible? This is possible by six pili, and that process is called conjugation. Got it? Got it?
Yes, ma'am. Got it.
And I told you there are two types of two types of pili. One is the longer pili. Longer pili helps in the sex. Okay, that is reproduction. And the shorter one, right? It helps in attachment. Shorter pili helps in attachment to any other rock. Okay, surface, any surface. Attachment of the bacteria, host tissue or rock, stone, whichever. And this pili is made up of which protein? Pillin. Next, you will find two types of bacteria. Okay, in bacteria there are two types of bacteria. That is gram positive and gram negative. Right? A scientist called Graham. Okay, he developed a technique which is known as gram staining. Okay, and how this happens is there is a chemical called crystal violet. If the crystal violet, how it happens, right? In this, when you take a sample, okay, in which there is many bacteria, okay, any water sample or something, okay, water, and what do you do is you put a crystal violet in there, okay, then you first you take bacteria, then you burn them by the stove, okay, the slide, and pick water, okay, then you burn it, then after that, if you burn, right, that bacteria will be fixed, okay, only so not totally burn, like warm it, the bacteria will be fixed, and after that you will put a crystal violet, and what will happen, the bacteria will all will be what, in violet color, all the bacteria we will be seeing violet color. Next, what we will do, we will next put iodine. What will happen, that violet color, right, it will stay. To fix the violet color, we are putting the iodine solution. Then after that, what will happen, when we put the slide in alcohol, what will happen, the gram negative bacteria, right, the coloration will go off. There will be no coloration after alcohol. If we put that slide in alcohol, what will happen, if it is gram negative bacteria, there will be no colors. And if there is gram positive bacteria, the color will stay. So what do? The color will be getting removed, right, by alcohol. So what do? We put saffronin. After putting saffronin, we will find some bacteria are in pink color, because of the saffron color is pink, right, reddish type pink, and the other one, crystal violet, was violet color. So you will be able to differentiate between gram positive bacteria and gram negative bacteria by gram staining. Got it?
Yes, ma'am.
1 minute, I'll show you beautiful diagram. Wait, gram stain. Okay. See this, right, when we do this experiment, we see, right, it is so beautiful that you see, wow, you tell wow. See, this violet color is what, which type of bacteria? Gram positive. The pink color one is what?
Gram negative.
Gram negative. Got it? How they are doing it?
Yes, ma'am.
So this gram stain. Now, what is the difference between gram positive and gram negative? So gram positive, they remain purple. They will remain purple after washing with alcohol. We have seen they do not retain color, right? The stain when washed with water. Here in the gram positive, the cell wall is single layer. Can you see? Single layer. And cell wall is bilayer. Here the peptidoglycan is 20 to 18 nanometer. So here it is thick, and here 8 to 12 nanometer. It is not that much thick, okay? Less thicker than gram positive, has more peptidoglycan layer, okay? Then murine, this peptidoglycan content is 70 to 80%. Here we'll find more peptidoglycan. In the gram negative, it is less. Then wall is smooth. Here it is wavy. Okay, it will contact with the cell membrane. Here in the gram positive, I have already told you 2 rings, S and M. Only one pair of ring will be present, that is S and M. And here the basal body will be containing 2 pairs, that is L, P, S, M. Outer ring and inner ring is present in gram negative. In gram positive, only one inner ring will be present. One pair of inner ring will be present. And the mesosome, we can find this right? We can see this, okay? It is prominent. We can see this. In the mesosome, it is less prominent. We are not able to see this, okay? Not able to see properly. Tell me.
Ma'am, can I use the washroom and go?
Yes.
Thank you, ma'am.
Okay. Sorry. Washroom.
Ma'am.
Yeah, you're back.
Yes, ma'am.
Are you able to hear me?
Yes, ma'am.
Your voice is cutting.
Ma'am, I'll check the internet.
Okay, and lastly, in most of the pathogenic bacteria which causes disease, right? Those bacteria belong to gram-negative. There are more gram-negative bacteria which cause disease than the gram-positive bacteria. Got it? Next, we'll see nutrition in bacteria. Okay. Based on the carbon source, energy source, and based on the hydrogen and electron sources, right? The bacteria are of 4 types. Okay, we'll see. First one, I'll write it here. Based on carbon source, nutrition in bacteria. First one, based on carbon source. So, if carbon requirement is met by carbon dioxide or organic carbon, 2 types. Let me see. Let me write. Only carbon dioxide, then it is called as autotroph, okay, for growth. And if it uses organic, bacteria uses organic carbon, then it is heterotroph. Next, based on energy sources, we have light phototrophs. If by oxidation of chemical compounds, from there the energy is generated, then either it would be the chemical compound, either it could be organic or inorganic, then it would be chemotrophs. Lastly, based on hydrogen electron sources. Here, if the electron sources is inorganic substance, then it is lithotroph. If it is from organic compound, then it is organotrophs. Okay, based on all these 3 things, based on carbon source, from where the bacteria are getting carbon, and based on the energy source, from where they are getting energy. Okay, and based on the hydrogen and electron source, we have 4 types of nutrition. Okay, first one, let me see this one. Photolithotrophic autotrophy, or you can say photolithotrophy. From this itself, you can see litho. First, from this name, we have to find source of carbon, then source of energy, then source of electron. Tell me now, litho, where we have seen litho? Here, right? Based on hydrogen donor. So, hydrogen here will be inorganic substance. So hydrogen here it will be, right?
Ma'am, I couldn't hear you for 5 minutes. Can you repeat?
You were not able to hear me. Okay,
Yes,
I was telling the bacteria nutrition is based on carbon source and energy source and hydrogen source. Okay, based on the carbon source, if the bacteria are getting carbon dioxide, that is autotrophs. Okay, if they are yeah, and if they are getting the organic carbon, then it is heterotroph. And next, based on the energy source, light energy, that is phototrophs. And if they get energy by oxidation, breaking of the chemical compounds, it could be either organic or inorganic compound. Okay, they are known as chemotrophs. And if the source of energy, source of based on source of hydrogen, we have inorganic substance. Okay, hydrogen, we are getting hydrogen from inorganic substance, that is lithotroph. And if we are getting hydrogen from organic compound, it is organotrophs. Now, from this, you can see it is given photolithotroph, right? So, first we can see litho, it is easier. Litho, we have seen from in which we have seen here in the hydrogen electron donor. So, the source of electron or hydrogen donor would be inorganic substance, right? Because name itself it is given litho. So their source of electron or hydrogen, what I have written, hydrogen. Okay, now it is correct. So inorganic substance. Next, photo. We have seen photo here. Source of energy is light. We have seen photo, litho. Then auto. That means source of carbon is carbon dioxide. Got it? Got it. So this type of bacteria, their source of carbon is carbon dioxide. Photolithoautotrophic bacteria, they have their carbon source is carbon dioxide, energy source is light, and the source of electron it is inorganic substance. Okay, so this is their mode of nutrition. Next, example of these bacteria is purple and green sulfur bacteria. Then also the blue green bacteria. Okay, they come under this category. Next, will you do the second one? I will give the name. Will you be able to tell the source of carbon, source of energy, and source of electron and or hydrogen? All right, photo, organotrophic, heterotrophy, or photo organoheterotrophy. Source of carbon, source of energy, source of electron.
The source of carbon would be carbon dioxide only, right?
Source of carbon will be carbon dioxide. Why? Is this autotrope? Here anywhere autotropes is given, photo is given, organo is given, hetero is given. So here source of carbon, will it be carbon dioxide? So what will be there? It would be organic carbon. Okay, organic carbon. Organic carbon meaning the carbon which is present in organic compounds. The carbon which is present in organic compound, we have sugar, right? There we find organic carbon. Okay. Next, source of energy. This one is easy, right? In this is given photo. So source of energy will be photo.
Sun?
Yes, tell me.
Sun.
Light.
Ma'am, in the light, can it be like any form of light?
Light meaning sunlight.
That's what. So only sunlight, no other light.
Okay.
Ma'am, what is the other one?
Other one was chemotropes. This, right, how do they get the energy source? They get energy source from organic compound or inorganic compounds. When they break down, when they oxidize, okay, from there the energy source we are getting. Such types of bacteria are chemotropes. Phototropes, they get energy from the light.
Okay.
Now here, is it any chemotropes are given? No. Only photo is given, so light. And source of electron, tell me. We have picked this photo. We have picked this hetero organic carbon. What is organo? So organo, if it is organo, then meaning?
Ma'am, it will be organic, not inorganic.
Yeah, it is organo.
Organic substance.
Organic
Compound, sorry.
So source of hydrogen and electron, it is organic compound. This one, did you get it? Now how we are naming?
Yes, ma'am.
How we are naming the bacteria based on the nutrition. Okay. Next, here the examples we have purple non-sulfur bacteria, then green non-sulfur. So non-sulfur, these bacteria will not produce sulfur bacteria. Okay. Third one, will you say? I'm giving the name chemo. Fourth one, right? What I will do is I will give the sources and you will name it. The other name we have chemo, litho, autotrophy. Tell me here, source of carbon, source of energy, and source of electron. Tell me, Priya, can you see here?
Yes, ma'am.
Tell me, source of carbon, will it be carbon dioxide or will it be organic carbon? Chemo, litho, autotroph. Tell me, what did you say now?
Carbon dioxide.
Yes, correct. Very good. You got it. Next, source of energy, chemo, litho, autotroph. This one we have taken, we have considered, done and dusted. Next, here, chemolitho, source of energy. Will it be light or will it be oxidation of compounds, inorganic or organic? Huh?
Oxidation of chemical compound.
Yes, good, very good. Oxidation of chemical. Next, so that's why it is chemo. Then litho.
Ma'am, inorganic substance.
Oh yes, the source of electron is inorganic substance. Very good. Ria, you're very good at this.
Thank you, ma'am.
Very good. Next, fourth one. I will give fourth one. We can do it like this way. I will give you source of energy, source of what is that? Carbon, source of electron. Okay. So source of energy, it is organic. I'll write in different color. Okay, or organic carbon. And the source of carbon, sorry, energy. Given, right, source of energy, it will be chemical energy, right? Oxidation for carbon, it is organic carbon, and for electron, it is organic substance. Tell me the name of this bacteria. Then how will you name them, this bacteria? How will you name this bacteria?
Ma'am, photo.
First we'll see carbon. If it is organic carbon, then it is, what do we name them?
Ma'am, chemo.
Hetero, right? Hetero.
Yes, ma'am.
Hetero.
Yes,
And the source of energy is chemical energy. What do we name? Oxidation. Chemo, right? Chemo. Then source of energy, a source of electron, it is organic. So organo.
Yes, ma'am.
So chemo, organo, heterotrophy. You got the name. You got the name. The other name is same, chemo, organotrophic, heterotrophic. Got it?
Yes, ma'am.
Easy, right? To name it is very easy. Just we have to know the source of carbon. And in the third, we have not given the example. I have not given the example. So here, example are nitrifying bacteria, nitrogen cycle, right? In that there are many, for fixing of the fixing of the nitrogen, we have nitrifying bacteria. Okay, so that is one example. And for the example of chemoorganoheterotrophy, here we have most of the non-photosynthetic bacteria. Got it? Everything here. Any doubt in this? Nutrition of this in bacteria? Nutrition in bacteria? Any doubt? Ria?
No, ma'am. Ma'am, I was wondering, will you share these notes?
Yeah, I will share. The thing is, I'm writing in iPad. Okay, the sharing would be done by the laptop. So in laptop, the what is this? OneNote. It should be synced. Then only I can share it. But the thing is, it is not syncing. Okay, it is not syncing. Okay, so when it sync, then I will share.
Okay, ma'am.
Next, we have the respiration. Tomorrow also we will do nutrition in bacteria in other ways. Okay, now we will see respiration in bacteria. There are two types of respiration, right? Aerobic and anaerobic. Aerobic and anaerobic we have already seen. Based on that, there are four types of bacteria. Okay, based on the type of respiration. Obligate meaning strict. Okay, obligate meaning strict. Aerobic, that means aerobic we know. What is aerobic? What is aerobic? What type of nutrition? What type of respiration? Aerobic respiration, tell me.
With oxygen.
So respiration in the presence of oxygen. That is aerobic respiration. Obligate aerobic, then this type of bacteria, right, what will happen is that they will live only in aerobic condition because they are obligate aerobic bacteria. So obligate aerobic bacteria, they require oxygen to respire. If there is no oxygen, that bacteria will die. So wherever there is oxygen present, then only that bacteria will be alive. So those bacteria which are completely dependent on oxygen, okay, those bacteria are called obligate aerobic. Okay, got it or not?
Ma'am, got it.
Next, facultative anaerobic. Facultative meaning adjustable. That means if they are given some other condition, right, they can survive. Anaerobic, usually the anaerobic. What is anaerobic respiration? Tell me. Anaerobic respiration. Anaerobic respiration.
Ma'am, respiration without the presence of oxygen.
Yes, respiration without the presence of oxygen. If oxygen is not present, right, they will respire. Okay, they will respire comfortably. Okay, very nicely. Okay, they'll be happy. Okay, content. But if there is no, if the oxygen is present, that time what will happen? They will be uncomfortable, but still they will respire. Okay, so that's why they are facultative anaerobes. They can live in presence of oxygen. These type of anaerobic bacteria, they can live in presence of oxygen. It's not like the in obligate anaerobes, what was there? In absence of oxygen, they die. In facultative anaerobe, even if oxygen is present, they can live. Okay, that's why it is facultative, adjustable. They are okay, I will adjust, like that type of bacteria. Okay, so they can survive both in anaerobic and aerobic conditions. Okay, next we have obligate anaerobes. These type of bacteria, what is their obligate meaning? Strict, without, I mean with, when oxygen is present, that bacteria will die. If it is without that surrounding, that wherever they are present, if there is no oxygen, they are happy, they can live. And if oxygen comes into picture, what will happen? Those bacteria, this obligate anaerobes, will die. Then we have facultative aerobic bacteria. These bacteria, what is there here? In this bacteria, they can live, okay, adjustable condition. When there is air, right, when there is oxygen, that time they will be very, very content, happy. Even if there is no oxygen, that time also they can adjust. They will not die completely. Okay, got it, Riya?
Yes, ma'am.
So let's see example of aerobic bacteria. We have Azotobacter. We have here Azotobacter. Facultative anaerobic bacteria, we have Azotobacter. You can see first one for the aerobic bacteria example was Azotobacter, and for the facultative anaerobic bacteria we had Acetobacter. Both are bacter, only aceto and aceto. Okay, then here in the obligate anaerobic conditions, right, the bacteria example we have Clostridium botulinum bacteria name. In facultative anaerobic aerobic condition, we have all the fermentation bacteria which do the fermentation. We have the fermentation bacteria, right, those all, except what? Acetoaceti. They are not what? They are not this acetoaceti. They are not facultative aerobic bacteria. They not written, not. Okay, so till this over, that's what here it is written. Can you tell me what is obligate aerobic bacteria, Riya?
Yes, ma'am. Ma'am, my internet.
Yeah.
Ma'am, I have to go for the next class. Can I-
Yeah, it will be at 6, right? You have 4 more minutes. Just
have
only one question. Oh, only one question. Obligate aerobic bacteria. What are they? Tell me.
Ma'am, they can only live, like they live in places where there is a lot of oxygen.
Yes,
and they
only
respire with oxygen. If oxygen is not present,
they will die. Okay, good. Okay, then you can leave. Bye bye.
Thank you, ma'am. Bye, ma'am.
Bye.